Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we are happy you made time to dial in today. And we will be talking about patenting strategies for small molecule drugs with Lucas Watkins and Alex Berne this morning. Good morning, you two.
Good morning.
Good morning.
And now, without further ado, I want to introduce our guests. So let's start with Alex. Alex Berne is an associate and registered patent attorney at Foley Hoag LLP, where he focuses on the preparation and prosecution of patent applications for chemistry-related inventions, with the majority of his work relating to small molecule drugs. Alex earned his JD at the University of Connecticut School of Law, and before his legal career, Alex spent over 3 years as a process chemist at Pfizer at the Groton site, and his chemistry there has been run on multi-kilogram scale in Pfizer's API manufacturing facilities. Alex also holds a master's in organic chemistry from the University of Pittsburgh, where he developed a novel Nazarov type 4-pi electrocyclization. And during his time as an undergrad at Boston University, Alex worked as a process chemist intern at Millennium Pharmaceuticals, which is now Takeda Oncology. Now, Lucas is a partner and co-chair of the patent prosecution practice at Foley Hoag. He advises on intellectual property needs, and including patent portfolio development and litigation strategies. And Lucas has extensive experience advising clients in the pharmaceutical industry, serving as patent prosecution counsel for global portfolios, protecting both marketed pharmaceuticals as well as development candidates. In addition to his counseling work, Lucas has litigated patent infringements and trade secret actions before U.S. district courts, international arbitration panels, the U.S. International Trade Commission, as well as the Patent Trial and Appeal Board. Lucas holds a JD from Harvard Law School and has a background in chemistry, biochemistry, and molecular biology. And he holds a PhD in physical chemistry from the University of Berkeley in California, where he used single molecule FRET to study protein conformational dynamics. So Alex and Lucas, really great to have you today. Really impressive CVs, and I'm really glad we have some people with law degrees here for the first time in the Flash Talk history. Honored to have you today with us.
We're really excited to be here. Thank you, Dennis.
Mm-hmm. That's amazing. And in our good old Flash Talk tradition, we always love to learn a little bit more about our speakers. So we're going to ask some warm-up questions. So what inspired you, and we probably will get started with Alex. Alex, what inspired you to start a career in the drug discovery field?
Yeah, I really enjoyed my organic chemistry class, especially learning the concepts of reactivity. And I found it really stimulating trying to develop synthetic routes based on those reactivity concepts. I also really liked trying to rationalize how unexpected products were formed based on those reactivity concepts. And I also found it really fascinating that some of the molecules that can be synthesized have incredible utility as drugs, potential drugs.
Yeah, totally. Lucas, how about you?
Yeah, for my part, I had already started my legal career. I was pretty early on when I realized that my chemistry background, in combination with the legal training I had received, would allow me to to help pharmaceutical companies get their therapies to market. And really, what's most exciting to me about that is helping small companies with a new compound make sure that they get the protection that they need, and you can really plan for the long term.
Amazing. Yeah, no, I think that's a very, very, very important, often underestimated part of the journey of a drug. And yeah, Lucas, staying with you, what's your favorite molecule and why?
So I thought about this a little bit, I'm going to say there are lots of candidates, but I'm going to say apalutamide. So the reason for that is it's a very patent-focused reason. So the drug enzalutamide was on the market, and then apalutamide was developed as it's a very close analog actually. But even though the structural differences are very minor, there were real differences in efficacy and potency, and those allowed the patent system to provide protection for apalutamide separate from enzalutamide, which I think is a really good example of how the patent system is really well designed for the pharmaceutical industry to protect these kind of seemingly very minor, but actually very significant changes.
Yeah, super interesting. That's actually one of my favorite cases too.
Ah.
Alex, how about you? What's your favorite molecule?
Yeah, I'd have to go with clopidogrel for a similar reason. So Sanofi had been developing a racemic mixture of clopidogrel and its enantiomer. They had progressed for 7 years and had even initiated phase 1 trials on the racemic mixture. But then, after extensive efforts, they were able to separate the enantiomers and found that clopidogrel provided all of the therapeutic benefit. And the enantiomer provided almost no therapeutic benefit and most of the toxicity. So then, because of the patent system, they were able to build a patent portfolio around clopidogrel and switch gears and move that through the development process.
Super cool. And I think we'll hear much more about this today
Yes,
as
yes,
well, right? And
yeah,
I did not know this story before, so thank you, Alex.
yeah.
That's super, super interesting.
Mhm.
Alex, staying with you, what are you most excited about in drug discovery today?
Yeah, well, I mean, I'm excited about all of our clients' work, but besides that, I'd say the research around targeted protein degradation I find really fascinating. So.
Definitely a ever expanding field of induced proximity. So I love that field too.
Mhm.
Lucas, how about you?
So, one thing that I see in the work that we do for universities is that there are, I think, a lot of new technologies being developed that are going to allow for really massive parallel profiling of, enzymatic activity for compounds, which is going to, I think, really reduce the burden of a lot of early biological testing and may help people focusing on developing clever analogs instead of just spending a lot of work on instead of spending a lot of time with biologists. I'm saying that to this crowd, which I think is mostly chemists.
Well,
But it
no,
is some
I
exciting
think
biology that allows that to happen.
yes, yes, yes. I think that's definitely something, in assay development, there have been multiple breakthroughs, and chemistry can work without biology. I mean, drug discovery is, of course, a big team sport.
Yes, exactly.
Yeah, and Lucas, staying with you, if you could go back to the beginning of your career and give your younger self some career advice, what would you say to yourself?
I would say, like, for me personally, it's keeping in better touch with all the colleagues that I've had over the years from grad school, from my, other jobs that I've had, because, sitting here today, my job is really about connecting people and finding ways to help people, either through my legal advice or through just connecting them with other people that I have. And that's something that, I do okay with, but I could have done a lot better with earlier in my career.
Very, very important part of the job, right? Networking.
Yes.
Yeah, sounds good. Alex, how about you?
Yeah, I would probably emphasize the importance of curiosity. I found that not only does it help with understanding something at a deeper level, but it seems like creative solutions seem to emerge from the process of being curious.
Yeah, I think that's great advice. As a scientist, I feel like we have some natural curiosity in ourselves, but
Mhm.
already instilling this in your children, I think is something that is really, really cool. Yeah. So last but not least, what do you like most about Drug Hunter, Alex?
Yeah, I mean, I really enjoy the Flash talks. I find it really exciting to learn about all the research going on, and also it takes me back to my time in industry. So.
Yeah, sounds good. Thank you. How about you, Lucas?
For me, it's the patent publications feature.
Oh yeah,
Yeah,
yeah.
maybe predictably. We do a decent amount of what I'll call surveillance for our clients, who are uh, working with a particular scaffold, want to see what's out there, and I think it's really great that you highlight some specific patent publications as they come out, see how the industry is developing.
Amazing. Sounds good. Yeah. Let me, on that, actually, thanks
Yeah.
for the great segue. I wanted to quickly show people how Drug Hunter works, or one quick feature of Drug Hunter, and that's the feature that Lucas already highlighted. So if you
Oh,
go here,
totally unplanned.
totally, actually totally unplanned. If you go here to the patents, then you can open that our patent database here. And what we do is we curate these patents weekly. So every week, as you know, patents publish on WIPO, and we go in there, and every month we extract approximately 200 to 300 small molecule patents that we then index by the patent number. So you can directly click on here and go to the patent, the month, the target, the therapeutic area, the originator, as well as an annotation. So here we actually go into these patents and extract the most exciting information here. How many examples are in there? What is some most exciting data in there in terms of biological data, DMPK data, in vitro or in vivo? So you can directly be informed and even search this annotation column here if you're specifically looking for patents that with compounds that have brain penetration or that have demonstrated efficacy in a specific model, you're able to really quickly search that. And also today, I wanted to very quickly teaser our drug discovery course. So we just released that yesterday. So if you go here on resources to our drug discovery course, you can actually sign up here if you're interested in learning updates. You can find we are going to roll out as a beta version beginning next year. All of the modules with their topics are already uploaded here. And you can kind of get a little sneak peek if you can click on introduction here. We have all those sections within the module. There'll be module quizzes. And if you click here in that first section, there'll typically be a video then beginning of January. And we have a little sneak peek into the slides that you will be getting from this drug discovery course. And with that, without further ado, I will now hand it off to Alex. And we will hear the presentation about patenting strategies for small molecule drugs. Super excited for this presentation. Alex and Lucas, stage is yours.
Great. Thank you, Dennis. So as Dennis mentioned, Lucas and I work at Foley Hoag. Foley Hoag is a leading law firm in the life sciences. We have a patent practice of over 50 patent professionals, including a large team of chemists that support our pharmaceutical clients with their small molecule assets during the preclinical stage, clinical stage, and commercial stage. We also have a large team of biologists that support clients with their biologics products. And then the firm also offers a suite of services to our life sciences clients, including advice on company formation, financing, and then navigating and complying with FDA regulations. And what presentation by lawyers would this be without a disclaimer? The views expressed are solely those of the speakers in the context of this discussion. No views should be imputed to employers, clients, or others. And with that, let's get started. So let's start with a quote from a famous patent scholar who says that the great emphasis in patent history is on the patent as incentive, the lure of reward to spur invention. And if there's one industry where the conventional incentive theory of patents is actually true, it is the pharmaceutical industry. So there's been an observation by several patent scholars that the pharmaceutical industry is special in that the patent system seems to incentivize innovation in the pharmaceutical industry to a greater extent than other industries. So we'll get into why that may be the case in a couple minutes, but first let's give you an introduction to patents. So patents, the patent right finds its basis in the Constitution, and some of the a theory on the justification for the patent right is that there's a grand bargain in which the inventor discloses their invention to the public, and in exchange they receive a monopoly right for a certain period of time. So patents are a legal right. They're the ability to exclude others from making, using, selling, offering to sell, or importing the claimed invention. It's not necessarily a right to practice your own invention, but rather a right to exclude others from practicing your invention. It's an instrument that can be sold or licensed, and the scope of protection is defined by specific claims. Notably, the right exists for a limited time, with the default expiration being 20 years from the filing date of your earliest non-provisional application. And in some cases, that term can be extended or reduced. So as I noted, your scope of protection is defined by your claims in the patent. So on the left, these are two patents covering inavolisib. On the left, we have a patent with a broader independent claim. So therefore, there's a broader scope of protection. You can see here that not only does this claim 1 cover the structure of inavolisib, but also related compounds falling into the scope of this genus. And on the right, we have a claim to a compound having the structure of inavolisib. So this is a narrow scope of protection. So broader isn't always necessarily better, because a broader claim may be more susceptible to invalidation if challenged, and we'll get into what some of the grounds for invalidation later in the presentation. Whereas a narrower claim, although it offers a narrow scope of protection, you may be more likely to survive invalidity challenges.
And I want to, I want to just
Yeah.
add to this, the generic claim, the independent claim that's on the left here, it's actually a pretty narrow example of a generic claim in a pharmaceutical patent. They can end up being quite broad, and the only, the two limitations are related to concepts that Alex is going to introduce a little bit later on. But basically, if you have a new chemotype that no one's ever explored before, you can write something that's very broad in a claim, and that's not going to be anticipated or but by other people's work that's already in the public domain. So you can see things like the heterocycles can be just described at a very generic level as being a heterocycle or a heteroaryl. Really, the sky's the limit, and what we try to do when we have really really new groundbreaking stuff is be very creative about the way that we claim something, so that we can have a claim broad enough to prevent competitors, follow-ons, so that anybody else who wants to operate in your space either can't or needs to have a conversation with you before they do.
Great. Thanks, Lucas. And also down here, we have a dependent claim. So dependent claims include all the limitations of the independent claim and then further limit that claim. So here, claim 4, we have the compound in claim 1, where in formula 1 is the chemical name corresponding to inavolisib. So why is it that the pharmaceutical industry seems to the innovation of the pharmaceutical industry seems to be so incentivized by the patent system? So scholars have theorized that certain factors may play a role, including very high R&D and regulatory approval costs, uncertainty in research outcomes, the ease in which competitors can reverse engineer new drugs, and drug manufacturing costs, which make drug molecules very vulnerable to imitation without the protection of patents. All right, so here's a graphical representation of the interaction between patents and drug discovery and development. So typically you'll start with drug discovery and preclinical testing, and during that period you'll file your first patent application. After you file your first patent application, you'll start to build your patent estate, and during that time you'll be able to attract investment, including licensing agreements, co-development partnerships, acquisitions. And then after the application publishes, may go up on Drug Hunter. Competitors may see it and be deterred from going into the field that you're researching. After FDA approval, then you start your patent exclusivity phase, and there are also regulatory exclusivities that interact with the patent exclusivity. And it's this patent exclusivity period that incentivizes innovation and justifies investment in drug discovery and preclinical testing, and justifies the massive expense associated with clinical trials and the whole drug development process.
One other thing about application publication, it may deter competitors. It may encourage them, depending on what they see that how good your molecules are. So that's just something to keep in mind, is that especially if you consider yourself to be in stealth mode, stealth ends when your patent application publishes.
All right, so when you file your first application is a really important decision, and it's very fact specific and depends on a number of factors. So the advantages to filing early is that you get an early priority date, which mitigates the risks associated with others filing first or problematic prior art publishing. And then once you start building your patent estate, you may attract investment earlier, and you also may be able to have earlier open and safe discussions with potential investors and collaborators. And it can be really dangerous to file after a public disclosure, so you really want to file before any public disclosure, such as in an article or presentation, because they can serve as prior art to your application. So sometimes you can remedy this under certain circumstances in certain countries. So the US has a 1-year grace period where you can disqualify prior art under certain circumstances. And then some other countries have similar grace periods. But there are many important jurisdictions like Europe and China that have no grace period. And then the benefit to waiting includes you have an opportunity to collect more data, which could support more robust claims, broader coverage, more relevant coverage, a greater range of subject matter, which provides flexibility to pivot your claims. And then you can also delay your patent prosecution costs, which can get quite high, as we'll talk about in a minute. And then you'll also have a later patent expiration generally, which results in a longer period of exclusivity generally. All right, so after you file that first application, what's the timeline? So your first application is typically a provisional application, which sets your priority date. And at that time, the application may be based on an idea and a work in progress. Notably, the provisional application does not publish, so doesn't necessarily become prior art if you decide to go in a different direction. So one option would be to file a follow-on provisional, and then let the first provisional expire, and it never publishes with prior art. After 12 months, you can file a PCT application. Sometimes you'll want to file a US non-provisional application at the same time. At that stage, you can update the application with any new data, and it's typically the last time to update the content of the application. It acts as a placeholder for filing in any of the PCT countries, and the International Search Authority performs an initial patentability search, and it publishes 18 months from your priority date, which may be prior art for future applications. And as Lucas noted, it'll inform your competitors as to what you're working on.
And just to note, the PCT countries, the PCT, which stands for Patent Cooperation Treaty, it covers essentially every commercially important country in the world. One notable exception is Taiwan, and there are a few other somewhat important but less notable exceptions. So usually a PCT application is sufficient, and I mean, certainly if you're looking for partnerships with Big Pharma, nobody will question if you only have filed a PCT application.
Great. And then after 30 months, you start your national phase prosecution, and this is where your costs can get pretty high because you're going to file in potentially many countries. The European Patent Office covers many countries at once, and then you get country-specific search and examination, in which the examiners determine whether the application satisfies the patentability requirements. In the US, those patentability requirements include utility. So does the compound have a practical use? Is it new? Is it non-obvious over what's been done before? And does it satisfy the enablement written description requirements, which are related to whether a person of ordinary skill in the art can make and use the claimed invention based on what's in your application?
Yeah, and so as Alex said, the 30-month date is very significant. Alex, if you want to just go back to that slide
Yeah,
for a
sure.
second. Because it's a big budget hit. You know, there are a lot of countries in the world, a lot that Big Pharma like to file in. Like Big Pharma can spend high 6 figures or even 7 figures on national phase applications. And you know, smaller pharma, you can file in a smaller number of countries, and still be very plausible licensing candidates. You know, I've seen very successful companies who just file their initial application in the US and Europe, and that's okay. But, you know, you should just know that when you file your kind of pivotal original provisional application for your chemical composition of matter, 30 months from then, you're going to have to make this decision. And to the extent possible, you should think about what countries you're going to want to file in, whether it's 2 or 10 is pretty typical, I think, for a small company, or more, and just make sure that you're budgeting for it. You know, when you're doing your use of capital for fundraising, you're thinking about that, because it's not something you want to be a surprise and only have a very small amount of time to prepare for financially. And most of them, by the way, those fees are government fees and translation fees. They're very, you know, it's very hard to negotiate those down. I'll put it that way.
Great. All right. Well, at the U.S. Patent Office, your application gets assigned to a patent examiner who's an employee at the patent office and trained in the technology area. So for a composition of matter case, it'll likely be a patent examiner with a chemistry background. And they evaluate whether the patentability requirements that we just talked about are satisfied. In almost all cases, your claims will get rejected in a first office action. And in response to that rejection, you can make arguments in which you find flaws in the examiner's reasoning, or you can make claim amendments to distinguish from, let's say, a reference that the examiner cited. And then once you file your response, the examiner will take a look, and they may allow the amended claims if you file claim amendments, or they may issue a new office action maintaining the rejection or under a new ground of rejection. So if after they allow the claims, your application can proceed to grant. You can also file continuation and divisional applications off of your initial application, and you can benefit from the priority date of your initial application. So for a composition of matter case, you'll often prosecute the compounds under one application and then file a divisional application to methods of treatment.
Yeah, and we have a lot of ways to either speed up or slow down the examination process. I would say the average, if you don't pull any levers, is going to be 3 to 5 years to go from an application being filed to having a patent. But you know, in some cases, you want to have that go very, very quickly. You know, we have pretty reliable ways of getting that done in 6 to 9 months. And by the same token, we can, you know, we can essentially delay things as long as we like at the patent office if your strategy hasn't been finalized and you just don't want to get the patent yet.
So after your first application, things typically don't end. So you can file subsequent applications claiming subject matter from later in the development cycle, which can be potentially very valuable because they can potentially extend your exclusivity period if you're non-obvious over the earlier applications. So here's an example where you'll have a composition of matter application along with methods of treatment. And then in some cases, you'll file a follow-on application to combination therapies, methods of treatment for dosages or additional diseases, and then further follow-on applications to salts and solid forms, pharmaceutical formulations, and other discoveries from late in the development cycle. And if you're developing multiple leads, you can build a portfolio around each of them. In some cases, you'll want to stop development of a certain lead compound, and then you may only file a fraction of these applications. And generally, the difficulty in getting the applications goes up the later you file them, but it may be worth it considering how valuable that extra time can be. So in summary, patents are essential for small molecule drugs, and you should be developing a patent strategy from day one. Doing so provides credibility with investors, and you can avoid mistakes early on that can really have significant detrimental downstream consequences. And considering how important your patent portfolio is, you should budget for it appropriately, and that way you won't have to cut your portfolio down as you continue. Great. So now that we're done with that section, let's unpack the patentability requirements in the US a bit more.
Let's pause for a second. I'm actually seeing a question on about Me Too drugs, and I think it makes sense to answer that here before we go on, because I think it's a really interesting one, about whether a Me Too drug, both the patent on the patentability side, whether you can get a patent on a Me Too drug, and also on the regulatory side, whether it can be approved. On patentability, a lot of that is going to depend. I mean, Alex is about to go into these, but if it's a minor change to an established scaffold and there's no advantage that can be shown to that change, then you may not be able to get a patent on it. And I think that's going to be a real, I mean, everybody in the field would know that and be able to predict it. So that's going to be a real barrier to proceeding with something that's strictly a Me Too. Usually, someone would be better advised just to do a generic, you've got to wait until patents expire, but you just sell a generic version of the existing drug rather than going through a whole development cycle for, what technically is a new drug that's not really going to have any added benefit. But if you can show superiority, you're very likely going to be able to get a patent. I mean, I think that in any drug that makes it to market, any new drug that makes it to market typically is going to have some reason that it is going to market, either, some superiority, it may be in, raw efficacy, it may be, in bioavailability and dosage form, something that's going to enable you to get a patent on it. Now, and I can't really speak in detail to the FDA standards. Maybe we can find another opportunity for one of these flash talks with one of our FDA lawyers on that. But I do tend to agree with you that we should be trying to sell drugs that are better than what's already on the market. That's all I'll say about that.
Great. All right. So let's start with the utility requirement. So the utility requirement is justified in that there's a desire not to have patents that are purely speculative. So this concept is captured by a quote from Justice Abe Fortas in the Supreme Court case Berne versus Manson. A patent is not a hunting license. It is not a reward for the search, but compensation for its successful conclusion. So in the context of pharmaceuticals, then, what do you need to establish utility? So let's look at the case of Inri Brana. There, the applicant claimed this chemical genus, which differed from known anti-tumor substances with the presence of this nitro group in the A position of this benzodiisoquinoline. Certain benzodiisoquinolines were disclosed in Paul, which is a reference that was noted in the applicant's specification. The applicant noted that compared to the compounds of Paul, the claimed compounds had a better action spectrum, and the compounds falling within this claimed chemical genus were also tested in vitro against human tumor cells and concluded that they had a good action. So the compounds of Paul were tested against 2 implanted murine lymphocytic leukemias, which were test widely used by National Cancer Institute. The claims were rejected because allegedly there was no specific disease against which the claimed compounds were active, was described in the specification. And then the tests offered by the applicants to prove utility were inadequate to convince one of ordinary skill in the art that the claimed compounds are useful as anti-tumor agents. The applicant appealed to the board, which affirmed, and then further appealed to the Federal Circuit, which reversed. So the Federal Circuit has a special importance in patent law. It's a special appeals court that has subject matter jurisdiction over all patent cases, exclusive jurisdiction, and also national jurisdiction. Oh, and what also makes the Federal Circuit unusual is that many of the judges have technical degrees, which is unusual for judges. The Federal Circuit disagreed on both of these issues. As to the no specific disease, they noted that the favorable comparison of the compounds to those of Paul implies utility against a specific disease, lymphocytic leukemia. And as to the second issue, they found that the in vivo animal tests were sufficient to establish utility. And they noted that requiring extensive proof of utility, such as treatment in humans, would disincentivize innovation in the pharmaceutical space because it would render the costs too high for many actors to enter to pursue their ideas. So in general, what we found is that you certainly want to have some proof of evidence of utility. Typically, in vitro data associated with the compounds that you're claiming is sufficient. But you don't need typically proof of human effectiveness in human treatment or even animal treatment. All right, let's move on to novelty. Is your compound or genus new? So what constitutes a novel pharmaceutical invention? So the issue of novelty is usually quite simple. So if you're claiming a genus and then a prior art reference discloses a compound falling within the scope of that genus, then your genus isn't novel and you'll have to amend your claim. But sometimes it gets a little trickier, such as with enantiomers, which is the subject of this next case. This case relates to clopidogrel bisulfate, which is the active ingredient in Plavix. This appeal addressed whether Claim 3 of the patent in suit, which is owned by Sanofi, is novel over earlier Sanofi patents that described the racemate. So as a little background, as I talked about earlier in our warm-up questions, Sanofi had progressed the racemate all the way to phase 1 before they were able to separate the enantiomers and found that clopidogrel provided all the favorable antiplatelet activity with no significant neurotoxicity, whereas its enantiomer produced no antiplatelet activity of virtually all the neurotoxicity. So then they moved forward with clopidogrel as their clinical candidate. And they also changed the salt from the chloride salt, which they had used for the racemate, to the bisulfate salt. So the question is whether this enantiomer clopidogrel is novel over the prior disclosures of the racemate in prior Sanofi patents. So the applied legal rules that the court used were claim subject matter is not patentable if it's anticipated, that is previously known, and the claim is anticipated only if every element and limitation of the claim was previously described in a single prior art reference, either expressly or inherently, so as to place a person of ordinary skill in possession of the invention. And this opinion was written by Judge Pauline Newman, who was a former chemist. The prior Sanofi patents disclosed the racemate and also recited some passages in the specification suggesting that maybe they described the enantiomers. As general statements, they said compounds having an asymmetrical carbon may exist in the form of two enantiomers, and the invention relates to each enantiomer and their mixture. Addition salts with pharmaceutically acceptable mineral or organic acids in both enantiomeric forms or their mixture. And then another patent stated that when the desired structure is obtained, it is isolated, and if desired, its enantiomers are separated and/or salified by mineral or organic acid action. So the Federal Circuit determined that these passages from the specification were not enough to render Claim 3 anticipated by these prior Sanofi patents. They noted that the reference must clearly and unequivocally disclose the claimed invention or direct those skilled in the art to the invention without any need for picking, choosing, and combining various disclosures not directly related to each other by the teaching of the cited reference, and that the disclosure of a genus in the prior art is not necessarily a disclosure of every species that is a member of that genus. And therefore, knowledge that enantiomers may be separated is not anticipation of a specific enantiomer that has not been separated, identified, and characterized. All right, let's move on to non-obviousness. So non-obviousness, obviousness rejections are typically the most complex to argue. So an invention is obvious if the differences between the claimed invention and the prior art are such that the claimed invention as a whole would have been obvious before the effective filing date of the claimed invention to a person having ordinary skill in the art. So often, as Lucas was hinting at with his answer to the question on Me Too drugs, a way to overcome these obviousness rejections is to have data showing that there's some unexpected property associated with your compound that you're claiming or your genus that you're claiming. For example, surprising potency or a surprising lack of toxicity. Yeah, so let's look at a case that
Yeah,
involves.
and I'll just say, because I think it's kind of amusing, legally speaking, the field of chemistry is considered to be an unpredictable art. And which helps a lot when you have to make an argument like this, saying, well, it was unexpected, that I got this improvement. I mean, it really is true in this respect. I mean, I think in a lot of other respects, the synthesis of a lot of compounds is fairly predictable, but that's not universally true, which is something that we take great advantage of when we're arguing about obviousness.
Thanks, Lucas. So let's look at a case involving aripiprazole, which is the active ingredient in Abilify. So the defendants in this case allege that claim 12 of Otsuka's 528 patent is invalid. So here's claim 12, which corresponds to the chemical name of aripiprazole. And the appeal addressed whether that claim was obvious over structure-related compounds that were previously disclosed by Otsuka. So as a little bit of background, the first atypical antipsychotic that was FDA approved was clozapine in 1990. Then risperidone was approved in 1994. And then between 1994 and 2010, the FDA approved an additional 7 atypical antipsychotics, including aripiprazole in 2002. But with the sole exception of aripiprazole, every FDA-approved atypical antipsychotic prior to 2010 has a chemical structure related to either clozapine or risperidone. So the court applied a special obviousness doctrine that's been used for pharmaceutical cases called the lead compound rule. And the opinion in this case was written by Judge Alan Lurie, who's another former chemist. So the rule is whether a new compound would have been prima facie obvious over particular prior compounds only follows a two-part inquiry. First, would a chemist of ordinary skill have selected the asserted prior compounds as lead compounds or starting points for further development efforts? The analysis is guided by evidence of the compound's pertinent properties such as activity, potency, and toxicity. Absent a reason or motivation based on such prior art evidence, mere structural similarity between a prior art compound and the claimed compound does not inform the lead compound selection. Second, would the prior art have supplied one of ordinary skill in the art with a reason or motivation to modify a lead compound to make the claimed compound with a reasonable expectation of success? And it is sufficient to show that the claimed and prior art compounds possess a sufficiently close relationship to create an expectation that, like the totality of the prior art, the new compound would have similar properties to the old. So the defendants put forth these 3 compounds that were previously disclosed by Otsuka as allegedly lead compounds. So the first compound, the untopsued butoxy, has a similar structure to aripiprazole but lacks the 2 chlorose substituents on the phenyl ring. And the second purported lead compound also has a similar structure to aripiprazole but has this propoxy linker rather than a butoxy linker. So for both of these compounds, they were disclosed among dozens of compounds in a prior Otsuka patent family. And that patent family also said that the compounds disclosed therein could treat a laundry list of diseases, including that they may be anti-schizophrenia agents. They also put forth this third compound, which is less structurally related to aripiprazole. Rather, instead of chlorose substituents, there's methyl substituents It also has this propoxy linker rather than the butoxy linker, and has this point of unsaturation here. However, this compound was actually a development compound, but it sounded like it wasn't going to go anywhere because it described the compound as having action that was not strong, and that patients receiving a 5 milligram dose experienced side effects characterized as severe. So the Federal Circuit disagreed that these were viable candidates as lead compounds. As to the unsubstituted butoxy and the 2,3-dichloropropoxy, the prior Otsuka patent family failed to tie those compounds to any meaningful suggestion of antipsychotic activity. And as to OPC 4392, those prior art articles that I just mentioned taught away from using this compound as a starting point for further antipsychotic research. And even if one did select this compound as a lead compound, the defendants failed to prove that the prior art would have directed one to make the various modifications necessary to convert this compound into aripiprazole. And the Judge Lurie concluded that the defendant's obvious case was a poster child for impermissible hindsight reasoning, which is when you use the disclosure of the invention in the actual patent or application to inform your reasoning as to obviousness. And further, that risperidone and clozapine, as of the priority date, were the natural and obvious lead compounds that you would have modified. And at the relevant time, there were no carbostyryl compounds that were marketed as antipsychotics or were publicly known to have potent antipsychotic activity with minimal side effects. Therefore, the carbostyryls were not plausible lead compounds, except in retrospect. All right, let's move on to...
I'll just say real quick, because I think the obviousness law, it really shows, I mentioned this earlier, how the patent system is set up for pharma and to allow these minor, seemingly minor improvements, like for apalutamide, to get a patent, which is what they need to be able to. You have to have a patent functionally to go to market, because otherwise you can't justify the investment. So I think this is why I think that if you have an improvement, whatever it is, but oral bioavailability, even if it's an improvement in being able to formulate something because of physical properties of a drug, you can usually get a patent that's going to justify taking a drug to market.
All right, let's move on to enablement and written description. So the enablement requirement satisfied by when the invention is described in such a full and clear manner as to enable a person skilled in the art to which it pertains to make and use the claimed invention. And part of the justification for this requirement is that grand patent bargain that I mentioned earlier, where the invention is disclosed to the public in exchange for the monopoly, right? So your invention does need to be disclosed to the public to receive that monopoly right. And then also, there's also notions of fairness. So did you actually invent what you're claiming? So let's look at the case of Idenix versus Gilead, which relates to sofosbuvir. So Idenix alleged that sofosbuvir infringes Idenix's patent, and Gilead admitted infringement, but alleged that the patent suit was invalid for failure to meet the written description and enablement requirements. So first, the Federal Circuit construed Claim 1 of the patent in suit. They noted that the preamble, which is directed to a method for the treatment of the hepatitis C virus, is construed as a narrowing functional limitation, which in combination with effective amount limits the scope of the claims to the use of some set of compounds that are effective for the treatment of hepatitis C. And as to the nucleoside, they construed it as having the following structure, where the methyl group is cis to the base. The court describes the methyl group as being 2' up, and the R1 and R2 can be any non-hydrogen substituent. So the main question is, did the disclosure in this patent would it have enabled a person of skill in the art to know which of the nucleosides falling within the scope of this claim are useful for treating hepatitis C. So the applied legal rules for enablement in, that the court put forth, the opinion was written by Judge Sharon Prost, where that a claim is not enabled when at the effective filing date of the patent, one of ordinary skill in the art cannot practice their full scope without undue experimentation. And in analyzing undue experimentation, the following factors in reruns are considered, which include the quantity of experimentation necessary, how routine any necessary experimentation is in the relevant field, whether the patent discloses specific working examples of the claimed invention, the amount of guidance presented in the patent, the nature and predictability of the field, the level of ordinary skill in the art, and the scope of the claimed invention. So applying these factors to claim 1, the court found that the patent is invalid for lacking enablement. They noted the quantity of experimentation is high, dearth of working examples, almost no guidance. The parties testified that the use of the modified nucleotides to treat hepatitis C was an unpredictable art, and that the scope could only support a finding of non-enablement. They also looked at whether Claim 1 satisfied the written description requirement and found that it did not. The written description requirement is distinct from the enablement requirement, and it requires that possession of the invention as the following dates sought, as evidenced by the specification, must be conveyed with a reasonable clarity to those skilled in the art. A genus can be sufficiently disclosed by either a representative number of species within the genus or structural features common to genus members so that one skill in the art can visualize or recognize genus members. And they compared it to looking for blaze marks for single out particular trees in a forest rather than simply pointing to trees. And the court found that the patent in suit did not satisfy the written description requirement as to Claim 1, and therefore claim was invalid on that basis as well. And that concludes our discussion of the patentability requirements. And thank you for your attention, and we're happy to take any questions.
Awesome, super. Those were very interesting cases of like clobetrogrel. I didn't really know that case, and also the preprazole case. Yeah, it's very surprising. And then the last case was I had heard of this, but the details I wasn't really aware of. So super interesting to get that teased apart. So yeah, for me at least, as a basically layman to this, it sounds very, very interesting. Some of these, and surprising to me, some of these decisions. But yeah, really cool. We had a number of questions by the audience. Lucas already took action here in the chat and answered a few of them. Lucas, do you want to maybe voice over some of those answers before we go into the open questions?
Yeah, well, I was thinking that, answering the open questions, I left some of the open ones because I thought they merited some live discussion.
Oh, okay, yeah, we can do that as well.
Great. But of course, if there are follow-up questions on the ones that I answered, happy to go into that as well.
Okay. Yeah, then let's go into the open question. So first question here from Ahmed Kamal. A biotech recently used generative chemistry to flood the chemical space around their patent lead compound, with basically essentially creating a chemical thicket to block fast followers. What is your take on this strategy?
So I have two responses to that. This is something that's very, obviously very, very of the moment. And one, I think it is a strategy that could work. I think that the main obstacle to it is that I think a fast follower, if they, there are so many different ways that things can go. Even disclosing 1,000 compounds may not block a really creative fast follower. Of course, you can disclose 100,000 with AI. There could be arguments that a fast follower could make, saying that, well, you didn't prepare these compounds. It's unpredictable how to make them. The dosing is going to be unpredictable. So getting on, the compound structure will be anticipated by that disclosure, most likely. Maybe not, but most likely. But there are going to be other inventions that a fast follower could could do, thinking about it strictly from the patent perspective. Now, commercially, are they going to be deterred and go try something else? Probably, until everybody starts doing this. Once everybody starts doing this, then nobody will benefit, right? But, one company doing it, I think you probably can deter someone from that. I think a major disadvantage of taking that approach is that you're eating your seed corn in a way, because your most likely backup compounds or next generation compounds are going to come from that same scaffold, and now you've just made it very hard for you to patent your own next innovations, right? So I think that the better way to do this, and this relates somewhat to a question that I answered also in writing, is to think carefully about what you've really invented. Think carefully about the way that your scaffold is docking, or the mechanism of action, and try to get a broad generic claim that, and that will disincentivize fast followers as well, without preventing you from then further innovating within that space. And so that's the way that I think I would prefer to address that with that concern. And fast followers have been a concern for a long time, and that's kind of a typical way to go.
Interesting. Yeah, super cool. Yeah, the race of AI is was already discussed, and it's a very interesting aspect for sure of evolving patent law. Yeah, and next question here. Under what circumstances would companies include just one compound structure in the patent claims?
I would say very rare circumstances. If you are iterating on a scaffold that's quite well explored in the art, you can think of a lot of common toxins. We'll talk about ADCs in a moment. Toxins that are used on ADCs that are, like everybody and their mother has come up with some new variant on a benzodiazepine, right? It's extremely hard to get broad genus claims in the benzodiazepine space. But at the same time, I have clients who have come up with, one additional tweak, right? You can still get improvements, right? In that case, you may end up getting a patent to one compound. Usually you would still have multiple shots on goal within an application, though. You would still try for a narrow genus. You would still try for more. So would I ever file a patent application that claimed only one compound? There wasn't like a formulation or something. Those you're always limited to one compound because the development is around the one compound. But in terms of a new chemical composition of matter, I've never done it. I don't think you probably ever would, because you're always going to want to have at least a few different options.
Yeah, totally. Yeah, and as you teaser, next question is about ADCs. So an attendee is asking, I recognize that today's talk is mainly focused on small molecule patents, but antibody drug conjugates are also a very active area. How does an ADC patent protect its small molecule payload, linker, and the final conjugate? And are there any notable differences compared to a standard small molecule patent that you could share?
Yeah, so ADCs are very, very interesting. We have a lot of clients that are developing ADCs, and much of like general ADC development is protected by what I would call small molecule adjacent patents. People may be developing new toxins that are designed for use in an ADC that you're going to protect with something that looks a lot like a small molecule claim. The linkers also, there's a lot of work being done on different linkers, cleavable or not, different levels of hydrophilicity. There are lots of aspects of linkers that are being studied, and those also, again, that's chemistry, that's not biology. So those look like small molecules, and then of course the antibody part of it is a more biological sequence-based. The real trick is how it all comes together. And this is one aspect in which I think patent law is not perfectly set up right now for like broad protection on ADCs. There's actually a case before the Federal Circuit right now called Seagen against Daiichi Sankyo, about the levels of disclosure that you need to make in an ADC related application. So this relates to the last topic that Alex was talking about, enablement and written description. So if you come up with a new linker, for instance, how many antibodies do you have to test that with to be able to get a broad claim to the linker itself without having to specify that there's a specific drug on one side and a specific antibody on the other? And in my opinion, the answer should be very low. And in other words, the benefits of the linker, the inventiveness of the linker usually does not depend on what antibody it's coupled to, because you come up with some new chemistry that's going to allow it to be cleavable in certain conditions or stable in certain conditions, right? But the patent law really hasn't yet caught up to that, and it's still a very uncertain area. One other thing I want to say about ADCs is that because you've got these three main components, you sometimes end up with three tracks of families protecting each one of the components. And you always want to be very disciplined in setting up your portfolio so that you don't create anticipation problems for yourself. For instance, when you're writing an application to a small molecule, if you know what your next backup compound or what your next area of SAR is going to be, you might keep that out of your earliest application, right? Because you don't want to just speculate and then create that prior art that, we were talking about a moment ago. And that's especially important when you're thinking about developing an ADC portfolio, because we see a lot of times, and this can happen when you're dealing with partners too, because often you'll license in an antibody from someone else, right? But if their antibody application, they have the sequences and everything that's typical in an antibody application. But then they say they have a very general claim that just says, and put that antibody into an ADC. That's a problem. That's a problem for me when I try to write a patent that gives the whole structure of the ADC. So that becomes something that patent offices look at for obviousness. It can limit the term of your patent, even if you still get the patent. So yeah, ADCs are very challenging, and they require a lot of thought to make sure that you're getting as much term as you can. Although they generally can be patented, but it's something that just requires a little bit of extra thinking.
Super interesting. Seems
That
like
was a
we need
long
an
answer,
extra
but it's a
flash
really
talk on
interesting.
that, right?
Yeah.
Yeah.
Sounds good. I think, in the interest of time, let's maybe two more questions. One more general question I want to end with, but first, this question here from Jason. We often encounter issues with quality of written experimental procedures in published patents. For example, incorrectly reported column chromatography conditions, etc. Have you seen any challenges to the composition of matter patents for failure of adequately enabling the synthesis of claimed compounds without excessive experimentation?
Yeah, that's a very interesting question. I haven't seen that. It's not often something that, you would have to do a lot of testing to prove that. I think if there was a really clear example of it, it certainly could be viable. Like if the main compound that's of interest, the drug, if someone ran the synthesis that was in the examples and it turns out not to have worked, and especially if you could prove that it was fabricated, I think that could be a very strong challenge to the validity of a patent.
That's good. Yeah, it's definitely interesting. Interesting to hear more about that. All right, let's end on a more general question for the both of you, and I think you are both the experts to answer this question. So, do you have any advice for chemists trying to transition to become patent agents? You both underwent this kind of transition, so maybe you can, both talk a little bit about what you would advise people on how to do such a transition.
Alex, you want to take this? I feel like I've been monopolizing.
Oh yeah, sure. So I think something you could do is study for the patent bar exam to see whether it's it'll give you an idea what the work is like and whether it's something that you may be interested in. Another thing you can do is take a position as a technology specialist and get some experience doing the work in that role so for technology specialist roles you don't need a patent bar number. So that way you can kind of feel out the role and see whether you like it. Yeah.
Anything to add, Lucas, or?
Yeah, I mean, just generally speaking, I mean, it's definitely helpful to come in with a reg number, but as Alex said, it's by no means required. Most of the people we hire are just coming either out of a postdoc or out of a position in industry with really no only interest and no experience or study in the field of patent law. We and every other kind of significant patent prosecution firm, we take that training burden on ourselves, and we have a whole set of internal coursework that we put people through, And I mean, what we're really looking for is a flexibility of mind, the ability to shift from, really a curiosity, investigational based career to something where your job is to advocate for your clients in front of the patent office and to translate that deep technical knowledge that you've already developed into something that can be digested by, an examiner who has a background but isn't, hasn't been doing this for 15 years, or a judge who has no background in it, or a jury. So that's what we try to look for. But it's a very fun career. I highly recommend it. You get to see a lot of different innovations, and you get to really help people bring important products to market. That's why we love it.
Awesome. Awesome. Yeah. Thank you again for this super interesting presentation. I mean, again, super surprising cases, for me, kind of unexpected outcomes, kind of the argument structure, learning like how to argue a specific case is for me like something super fascinating. So really thank you for presenting these super interesting case studies today. Thank you to the audience for all the engaging questions. And if you have any follow-up questions, please email Alex and Lucas. They have their email addresses are in the slide docs that you will see on YouTube. And with that, again, another round of applause for Alex and Lucas from everybody. And yeah, we will see you here again on November 20th, and we will talk about process development for lenacapavir, which is also a super interesting drug from Gilead. We saw Gilead already famously featured for sibospovir today. So next time we'll see the process route for lenacapavir. So with that, I wish everybody a wonderful rest of their week, and we'll see each other at the next event. Thank you.
Thank you.
Everyone.
Bye now.